So let me start first of all by welcoming all of you to the first Leading Lights lecture of this year is particularly special because it's our 20th birthday. So not only is Anne trailblazing as the first digital Leading Lights lecturer, this is also the very first lecture during our 20th birthday celebrations, which will be a year-long thing. So despite all the challenges with our current circumstances, we want to try and keep things as normal as possible. So we will be doing all of this. All our teaching will carry on as normal. We'll try to keep all the research going, and very much part of all of that are events like this where we come together and listen to outstanding research by a colleague, etc. So a little bit about Anne. So Anne's got fantastic pedigree when you look at her CV, and in fact I looked at it more closely just before this lecture. So she's been to good quality institutions like the Max Planck, the Wellcome Center for Cell Biology in Edinburgh, the Marie Curie Research Institute. But of course, a high point in her career was when she moved to Warwick Medical School 10 years ago. So it's exactly a decade now. And during that time, of course, she's progressed in her career so that she's moved up. And this is something I think we should be really proud of. That is, we have an environment in which people make excellent progress in their career. She became a professor, full professor last year. And again, achievements at Warwick include a Lister Prize Fellowship and then a Wellcome Senior Investigator Award, etc. So, and she's also contributed significantly to teaching. Initially, our MRC doctoral training program, organizing the taught element there, etc. So I think she's now been playing a leading role in several of our activities, and I think I'm particularly now pleased to be able to hear her about her work from her. So Anne works on microtubules, understanding more about the mechanochemical processes around that drive cell migration in particular, which is relevant to conditions like cancer. And of course, that's what she will tell us, and hopefully a bit more about her personal journey up to this point. I'll be very interested to hear about it. I'm sure everyone listening in will feel the same way. Before I invite Anne to present her lecture, can I, one or two housekeeping things? If you have not already done that, could I ask you to mute your microphones? It'll make the audio much better for everybody. Second, it's also a good idea to switch off your video because I think all eyes should be on this, on Anne and her slides rather than on any of us. So I'm going to do the same thing. Please don't go off and make coffee or other stuff. Please do listen to the lecture because that's one thing we can't see what you're all up to. So I think I think it'll be a really good lecture. So I would now like to invite Anne to give her lecture, please.
Thank you, Satish, for the introduction. Not setting any expectations here. Okay, let's see whether we can do that remote thing here. Great, I'm seeing none of you. I hope you see me. Yeah, it's very weird to give that lecture not in front of all of you and seeing you in a room and maybe celebrating afterwards, but instead sitting in my lonely, messy office. But anyway, here it goes. So my lecture is about mapping the intracellular transport network and what my lab is up to since quite a few years. So the problem really is looking at how cells organize their inner space and how they get components of the cell from one place to the other. And this here is a view from space onto Berlin. And you can see really nicely how the roads line up. And this is kind of the infrastructure network that humans have built, right? And
I'm sorry, Anne, sorry to interrupt. Your slides aren't changing.
My slides aren't changing? They are not? Why are they not changing?
We can only see your titles. Ah, now they've changed, yes. Sorry, carry on now because it is changing.
Yeah, but now I'm no longer in my preview. If I go to full screen, you can't see them anymore.
Try that again.
Is something changing now?
No.
Ah, why Andrew's coming troubleshooting now?
You're not full screen. So
You
share your desktop. You share your whole desktop.
Oh, and we need to share my desktop. How am I unsharing? Okay, wait, let me unshare.
Yeah.
Let me unshare my screen, and then I need to share my whole desktop,
Yeah,
supposedly. What screen 1? Okay, let's see. And from there, I'm trying to do that. Let's see.
Yes, I got the title slide again.
Title slide, and you see it changing?
Yes, it is changing. Excellent. Yes, sorry, I won't interrupt anymore.
Yeah, perfect. That was Andrew troubleshooting from next door. Okay, perfect. Good. So it's good that we still have neighbors in our offices. Okay, sorry. So this is the transport. So this is Berlin from space. Sorry, you haven't seen that when I talked about that earlier, I assume. And this is all the roads that kind of we built to transport. And cells are very similar in the sense that we have that road and or rail network as you once saw. And those are the infamous microtubers. And this is the kind of the network and the road network that we are studying. And of course, if you look at Berlin, we actually are not sitting in space. We can actually go down to Berlin and we can figure out what's going on. And we have very clear maps where we know what all the different transport networks are. We know where we have rails, we know where we have roads, we know where we have rules that you can only go one way, and the way how things are transported along those roads. So we can, of course, go directly to the city and we can see, we can observe how each of those vehicles is moving, at which speed it moves, and what is it transporting. That is a bit more difficult to figure out in cells because we can't really go to that level as we can go down into Berlin, but instead we kind of see things more or less like from outer space. And those are the key questions that we still want to answer, basically getting to a view of observing what is happening. So how are those roads built? What are the traffic rules? How is the layout of that network determined? What does each of the lorries or whatever you call those transporty mobiles is transporting? How are they controlled? And then, of course, related to that, of course, how are these processes hijacked, for example, by pathogens who are using that transport network to kind of also go to the spaces they need to go? And how are they deregulated in diseases? And before I really go to showing you how we answer those questions, I thought I go back, as Sudesh mentioned, and show you the journey, how I kind of got to that point of asking these questions and how I got interested in those. And I start quite far way back when I went to secondary school in this not a very unassuming building, actually. And that was in Eastern Germany, here in Halle. But it was a great school in the sense that I got one of the best educations in math, natural sciences and technology one could get in secondary school. And conveniently, by the time I'd finished school, that border here had vanished. And I actually was free to kind of move out of that space and start my kind of journey over that map. So that was the time. So we now have 1994. Molecular biology was really coming and going. And there was this great undergraduate course in Hamburg, which was teaching biochemistry and molecular biology. So really the newest techniques, how you could interrogate how life really works. And for my final year project, I chose to work on this plant pathogen, which is causing kind of that disease in barley. And we were trying to find some virulence genes in that pathogen by kind of mutating it. So after my degree, I decided to do a PhD, and I actually happened to stick with the theme of a plant pathogen. So I moved to Munich into the department of Regina Karmann and worked with Gero Steinberg, who were investigating this pathogen that affects maize and makes those big tumors there, which are actually a delicacy in Mexico. So not all pathogens are only doing bad stuff. But we were not so interested actually in the virulence in that case, but we actually studied more that pathogen itself, which when it propagates is being that kind of elongated shaped budding yeast, Ustillagomades. And this was the time, so now it's 1999, that is 5 years after GFP has been cloned for the first time and seen that it actually works in cells. So we used that to do some really nice fundamental cell biology with it. So we also had the first genome of Ustilago, so we could start tagging components inside the cell and actually see for the first time how they looked like. So here you see examples of how the microtubules look like in Ustilago. We could see that they are dynamic, we can see them shrinking, we can see them growing, we could see how motors are moving and buckling those microtubules, and we could also label subcomponents. Like here we labeled the on the left hand side, we label the plus ends of the microtubules, how they are growing, so we could observe the polarity. We could figure out how during the cell cycle the microtubule cytoskeleton is changing and supporting the propagation of that organism. We also looked at microtubule motors. So here you have endosomes labeled and microtubules, and you can appreciate how those vesicles are zooming along the microtubules inside those cells. Here we depleted the two genes that make up together the dynein motor, and we could observe phenotypes like nuclei were accumulating in cells, so they were not segregating properly. And we could observe directly how this happened. So dynein was important for moving the nucleus from the mother cell into the daughter cell at the onset of mitosis, and at the same time also moving actually DNA outside of the nuclear envelope and leaving that behind. And of course, because that process is not happening when dynein is depleted, the nuclei just got stuck in the mother cell and never kind of made it to the daughter, so the cells could not proliferate anymore. We also found that dynein is involved in actually organizing microtubules. So you see here in control cells that the microtubules are nice and straight and long along the long axis of the cell. And here we have a temperature-sensitive dynein mutant in which you see that after a couple of hours into that non-permissive temperature, you see the microtubules are a mess. We also studied kinesin, which is also a motor that walks along microtubules usually and transports cargo, but we found that that also is important to actually organize microtubules. So here you see why microtubules are called microtubules. So this is a cross-section in electron microscopy, and you can see that those are little tubes that are running along the cell. So this is a longitudinal view of them. And we could observe that when we depleted that kinesin, or deleted actually, then we reduced the number of microtubules that occur in bundles, and we could also see that in the absence of the motor, microtubules are no longer moving as much, and if you overexpress the motor, we had a lot more motility in microtubules. So those motors are actually not only transporting cargo along the microtubules by walking along it, but they were also organizing the microtubules. So after that, I decided now it's time to kind of work on some proper cells. So I started my postdoc at the Wellcome Trust Center for Cell Biology in Edinburgh. And I got a fellowship from the German Science Foundation to do this, and I worked in the lab of Andreas Merdes. And Andreas was really interested in the centrosome, and he had started to work on skeletal muscle cells because they are known to kind of lose the centrosome once they start to differentiate. And I came and I was really interested in how microtubules are organized, so he let me do that in his lab. And microtubule reorganization really is spectacular in muscle cells. So you can see here that we have this radially organized cytoskeleton in those myoblasts, so they are undifferentiated. They are migrating round. So you can see that here because the kind of the centrosomes, of where most of the microtubules are organized, are kind of leading the nucleus, and then you have lots of microtubules towards the front and less so towards the back of the cell. But once you trigger those cells to differentiate in culture, you can see how that centrosomal focus of microtubule organizing proteins is dissolving and instead is relocalizing around the surface of the nucleus. And you get this paraxial microtubule arrays forming. And then several of those cells align with their kind of tips with each other and fuse in order to form those myofibrils. And this is how you form a long, nice myofiber. So this was really a brilliant model to study microtubule reorganization, because you go from this radial kind of centrally organized array to those arrays where centrosomes no longer play a role. And I initially studied those proteins that give you those beautiful fireworks in cells, those EB proteins. They bind the ends of growing microtubers, as you can see here. So they're tracking each microtuber as it grows in cells. And mammalian cells actually express 3 of those. And it was very clear that in those muscle cells, they are differentially regulated. So one is the ubiquitous protein, EB1. EB2 is kind of shut off very quickly once cells start to differentiate, and EB3 is really upregulated strongly. And I could show that EB3 is really important for the differentiation of those cells. Firstly, you can see here the tips of the cells really at high magnification. You can see microtubers and growing and shrinking and touching the cortex, and they get really very stabilized at this cortex by being kept in that dynamic stabilized state of being kept near the focus for several minutes while still undergoing growth and shrinkage. And if you depleted that plus-end tracking protein that organizes a whole complex of accessory factors at the end of the microtubers which fine regulate the dynamics of microtubers, then we could see that a lot of that goes wrong. Microtubers just kept growing when they hit the edge and kind of are buckling in the back. And they very quickly shrunk away and kind of got lost from this edge. And that resulted in those cells no longer being able to elongate properly nor to fuse. So here you can see in the control depleted cells some nice long cells with loads of nuclei. And here you can see that in the background that is still happening, but those cells where we depleted that EB3 protein, they can no longer take part in that. I also observed the organization of microtubers and their motility, for example, using that experiment, using photoactivation, because the microtuber cytoskeleton is really very dense. So it's no longer possible, like in Ostilago, that you could see all the 3-4 microtubers it had. Now, suddenly I was working with cells with a super dense network that is packed full of microtubers, but with that little trick of just labeling bits of microtubers, you can actually use that, as you can see here, if you then film them, to observe the motility and the reorganization of those microtubers. And you can actually see that at second scales, microtubers are reorganized very rapidly. Not only do they grow constantly, as you could see in that movie I showed you earlier with the EB proteins, that they're being built and disassembled, but they also get moved around by motors constantly. However, once those cells differentiated into myotubes, their movements were much restricted. So you can see occasionally a microtuber moving out parallel and slowly, but they're much more well behaved, which I thought was very intriguing how that happened. The motors that are important in moving those microtubers, because if we take them out of the cell, we see that the microtubers are hardly moving anymore, are like I showed you before, actually, in that basic fungus, were dynein and conventional kinesin. So both of those motors being also involved in those muscle cells in moving microtubers. But once those cells differentiate, the motors are still there, and the motors are still important to transport organelles, to position the nuclei, and do other important jobs. So at that point, I actually hypothesized that in those differentiated cells, there must be some other players that are kind of cross-linking those microtubers and kind of holding them in place and prevent those motors to kind of causing havoc and moving them around like crazy. And armed with that hypothesis and some other ideas, I applied to start my own lab, and I was happy to be taken into the Marie Curie Research Institute in Oxted, and started there my lab to investigate cytoskeletal organization. So that was down here in Surrey, and the views from there were absolutely spectacular. So I thought I need to show you those. So soon enough we started. I had brought along my research assistant and hired my first PhD student, Binyam. And Binyam investigated that hypothesis. And we somehow had this idea that MAP4 protein might be a really good candidate for being that cross-linker. Don't ask me why, but it turned out that we seem to have been right with that. Again, every good thing comes in 3, so muscle cells expressing actually 3 major isoforms of those MAP4s, in ubiquitous form, and a muscle-specific one, and OMAP4, which was the new isoform we kind of discovered somewhere in some databases, and nobody had ever looked what that was doing. And it turned out that OMAP4 really was important here. If we took out each of those 3 proteins from muscle cells. OMAP4 was the one that was required for cells to elongate. It was required for them to fuse, and it was also important to organize microtubes nice and neatly. Similar to the assay I showed you earlier, this is looking at the motility of those microtubule bits that we activated. When we depleted OMAP4, we could see much more movement. So we could see this looping round of microtubules again. And that seemed to really be that we had identified that factor which holds microtubules kind of in check in differentiated cells. The other great thing about the Marie Curie was meeting those guys here, and I hope you still recognize them, even though some don't have as much hair anymore. So there was Andrew, who had started his lab a little bit before me, who was always only thinking about dividing cells and did not care about the 99% of what cells did the other time. Good, he can't say anything right now. And Rob and his Motors lab, who were really interested in figuring out how kinesins really work in all detail. And together we kind of formed this microtubule dynamics interest group at the Marie Curie. We had joint retreats and we had a lot of fun. And then there came this day, not actually that long, in about 2 years after I started my lab, when the Marie Curie Cancer Care decided that they no longer wanted to support that research institute in Surrey, but instead ship us off somewhere else to some universities to do our work there. But they allowed us to got some funding with it, and of course take all the equipment that we had. So the next few months, I think Andrew, Rob, and I spent a lot of time in Cafe Nero and Oxted to discuss what we're going to do with that opportunity, or with that disaster turned opportunity. And we came up with a plan that involved setting up this Center for Mechanical Cell Biology in Warwick. And surprisingly, what was born in the Cafe Nero and Oxted seemed to kind of come reality. And so this is kind of early days when we had more or less just arrived at Warwick, and I guess we have gone from strength to strength. And I guess you can see what is of the seems to be right now all around you. So going back now to those questions, I think we have arrived more or less at Warwick and in the presence now. So I want to talk a little bit about how we are trying to answer those questions and what are we doing about them. So we set up our lab in doing kind of a wide variety of techniques in order to address those on different levels. And one level is of course the level of the cell, where we're using those markers like I showed you earlier, those EB proteins that are tracking the growing ends of microtubules, and computational analysis in order to kind of track those and rebuild kind of those networks in our computer. So to map out really where the microtubules are growing, in how fast they're growing, and in which direction they're growing. And also in those muscle cells, which are kind of crazily dense, we are using super resolution techniques, or in that case, this is expansion microscopy, to really be able to kind of trace the filament network, and again, fitting snakes to them, in order to kind of map out how microtubules are oriented and organized. And when we then remove those players that we think are interesting and involved in those processes, that we can see changes and measure those changes objectively. So that is one of the kind of approaches that we are using. And the second approach to study transport in cells is that labeling the cargo is a pretty obvious one. So this here is a cell where neuropeptide Y is expressed, and you can see little vesicles that carry that cargo up and down the cell. And the interesting thing here is that usually people have to believe that you have a cargo, and that cargo will hook up to a motor, and there are two types of motors that are kinesins that kind of move anterograde to kind of the tip of the axon or outside of the cell, and then you have the dynein motor that transports to the inside of the cell. But we actually observe that cargos tend to move in both directions. And if you pick one out here, if you manage to do that, you might manage to see that it reverses direction. And you can also see that in those chemographs that you can see in our data a lot, where you basically draw a line here, and then we plot that line over time. And that allows you to really see, so diagonal lines are movements into the right direction, so the tip of the cell, and diagonal lines in the other direction is moving towards the cell center. But we can also observe reversals in the movement of those cargos. And those are very intriguing, and people are believing now that most cargos are actually recruiting motors of different polarity. Also, we don't really understand how that then not just results in a tug of war, so where both motors are fighting with each other and you actually don't make any progress. We actually observe in cells quite the opposite, that when we deplete one motor, that the cargos don't move at all anymore, rather than collecting on the other side. So that suggests that there is some synergy or codependence of motors working together. And one way how we envision that this codependence could work is by those motors acting in joint complexes in which they can regulate each other. And they have actually physical contact to switch each other on and off. And one of those complexes we are currently very interested to work and working on is the one that is made out of dynein, dynactin, and the kinesin KIF1C. And this is kind of held together by a cargo adapter hook 3 in that case. So doing cell biology experiments, we can take out components and see what happens. But really the biggest impact, I think, in terms of technological development in my lab was in recent years that we started to do those in vitro reconstitution assays. And this is here one example where we do one of those assays. So we attach a stable part of the microtubule that is in this movie here, the kind of the dark bits that you don't see. So we attach those to the surface of the glass cover slip, and then we put purified tubulin protein in there, and that grows then those extensions of the microtubule. And each microtubule has a fast-growing dynamic end. It would be in that case this one here. You see that grows much faster, and a slow-growing, less dynamic end. And one is the plus end, and one is the minus end. So in those assays, we can then study the dynamics of microtubules. We can add factors in and out at different quantities, and we can see whether those microtubules assemble faster or maybe assemble slower, and maybe more often undergo those switches between growth and shrinkage, as you can see here. So usually microtubules have extended phases where they either grow or shrink. And we can also observe what happens when two microtubules meet each other. And again, add factors that might modulate what happens. So this is really super powerful in really trying to understand the mechanisms of how this works. So we use that, for example, to reconstitute this plus end tracking behavior that I showed you before, where you have those comets shooting out on all growing microtubules in cells. In vitro, you can reconstitute that by, again, growing microtubules on the cover slip surface, as I showed you, and then adding that protein that we labeled here into your assay. And you can see that that is all that you require in order to mimic that behavior. So you see that those bright comets are forming at the ends of growing microtubules, and the moment that microtubule stops growing and shrinks, that signal is gone, which is exactly what happens in cells. So we use that to study the properties of those proteins and how they regulate microtubules. We also use that assay, as I alluded before. So now we are adding this OMAP4 that I told you we identified as a microtubule organizer. And we can see now in those assays, when now two microtubules are meeting, like those two are coming reasonably close together, that OMAP4 is zippering them up and organizing them into an antiparallel bundle. And as we know in that system, all the orientation of all the microtubules, we can figure out that it has a preference for antiparallel microtubules, and we can really probe the properties of that reaction, which is something that is almost impossible if you wanted to do that in cells. Other assays, similar, maybe less complicated in some ways, microtubule gliding assays, where we use those motor proteins I talked about that can walk along motors and transport cargo, and we kind of reverse it by attaching them to now the cover slip and add microtubes on top. And then something like crowd surfing happens, where those motors on the surface are kind of trying to walk along that microtubule, and they actually end up transporting those microtubules over each other. Not only are those assays beautiful, but you can also test some ideas using those. And one of the ideas we tested, again, going back to OMAP4 that you already know about, the idea was, as I said, our hypothesis was that that motor would be cross-linking and preventing motors from driving microtubules around in cells. So, of course, it needed to be strong enough in order to prevent kinesin motors to kind of pull those bundles apart. And that experiment is showing that. You can see in the background all those single microtubules being driven around on the surface of kinesin, but that bundle that is bundled with OMAP4 is making no progress because it's an antiparallel bundle, and there will be motors trying to pull one microtubule that way and the other microtubule that way. And as they are held together by OMAP4, it's going nowhere. So this is an example where we can use those assays to really test that idea whether this bundling protein is strong enough to hold microtubules together and prevent that sliding. And finally, or maybe not finally, we use single molecule assays where we stick the microtubule solidly on the surface and then add single purified motors which carry a fluorophore, and then we can see them running along those microtubules. And that, of course, allows us to study its motile properties when it's not under load. And we can also learn quite a few more things. So this is an example where we use dynein. And dynein on its own tends to be in this state of being auto-inhibited, where its two motor domains are kind of pointing in opposite direction and it can't really go anywhere. And if then it might only do some small excursions or maybe even just diffuse. And usually dynein needs have a cargo around that binds dynein in order to sort out its 2 feet, and then it can start walking. But we found out relatively recently that dynein can actually cross-bridge microtubules. I mean, we knew that already. I showed you earlier images in cells that dynein is really important to slide those microtubules around and organize microtubules. But we actually found out by doing those single molecule assays on single microtubules versus on bundles that were formed by dynein, that dynein now suddenly could actually move processively on those microtubules. So on a bundle, dynein suddenly becomes a fast and processive motor. And the idea is that dynein is actually doing that by cross-bridging 2 microtubules, like a climber would be when they are kind of chimneying up a rock face, which is a very efficient way of kind of making the way up. The same dynein is spreading its 2 motor domains on those 2 microtubules, and that helps to disentangle those 2 motor domains that would otherwise go into this auto-inhibited form. Another thing we are using those single molecule assays for is to study this cargo activated activation. So this is an example here of that kinesin KIF1C we are studying, and the cargo adapter hook 3, where you can see that if the motor is alone, we have much fewer events of motors landing on the surface because they are in solution in this auto-inhibited dimer form. But if we add that cargo activator or the cargo adapter and activator at the same time to our assay, then we have a much higher rate of motors landing on the surface, and then they move together with that cargo adapter on the microtubules, and that looks then a little bit like that. And we also have been recently successful in using that to reconstituting that bidirectional transport complex that I introduced to you earlier. So I've just shown you that part of it where KIF1C binds to hook 3 and that activates the kinesin motor. But it had already been known, and we are showing that here, is that hook 3 can also activate dynein. So I showed you earlier that dynein on its own is not a processive motor, and you might get very brief and maybe diffusive motion out of it. But if you bind hook 3 to it and dynactin, then dynein-dynactin hook 3 becomes a very processive complex. and that accumulates on the minus ends of microtubule. So it goes in the opposite direction of kinesin, as you can see here, that goes to the right-hand side. So if you put the whole thing together and we actually form that bidirectional complex, then you can see that we have now kinesins moving the wrong way around. So they move to the minus end together with HOOX3 and dynein. And we also see dyneins going to the plus end, like here, oops, here. And there's another example of here, because it's transported by KIF1C in that complex. And what we do not see when we reconstitute that complex is that there is a tug of war. We don't see a dramatic amount of static, slow events where both motors fight to each other. And we are currently trying to figure out why that might be. But we seem to now have something in our hands where outside of the complication of the cell, we can actually figure out how that motor codependence is working. And I think that might be my finally technical slide here, is that we are collaborating with Rob Cross's lab using the optical trap that Nick Carter has built. And that allows us to probe the forces that single molecules make. So we have a big plastic bead on which we hang a single motor, and then we can see how that single motor is pulling the bead out of the kind of center of that laser trap. And you get traces like this, where you see this is the laser trap center, and you can see how the kinesin pulls the bead out and reaches a certain force, and then eventually kind of it will fall back to the trap center. So we can use that to determine the forces a single motor can make. And we use that also to study mutations that in KIF1C that we find in human patients with hereditary spastic paraplegia. So those patients have a severe range of symptoms. It's usually a complicated form of spastic paraplegia with also some cerebellar dysfunctions. And the idea was that those motors are for some reason weaker. When we put them in our single molecule assays, we still find that those motors can move and they actually still accumulate at the plus end of microtubules as the wild type motor would do. But if you put them under load into that trap assay, you can actually see that those motors have a dramatic reduction in the forces they can produce. That motor really, we can't see anything happening in terms of being able to pull that bead out of the trap. So that gives us a handle to really understand the biophysical parameters of those motors that are implicated in disease. And we are now trying to also take this the other way around and trying to generate a disease model to study kind of those motors in the in vivo context and try to kind of bridge the biophysical properties to the outcome in a neuronal development. See, it's never the final one. Then we have this beautiful lattice light sheet microscope, and there's one example I want to show you what we use that for. And this is an example where we study how the microtubule cytoskeleton is hijacked by a pathogen. So here we study Marek's disease virus, which is of really important, economically important, because it affects chicken, and poultry is really one of the major meat sources nowadays, and growing so. Marek's disease is very efficient killing chickens, and usually all chickens are vaccinated against it, but there is a risk that those vaccines will not ever work. So we are studying that virus and how it's being transported inside cells. Interesting that virus can only affect other cells by moving to the surface because it's cell associated, so it will not lyse the cell, but it will actually go to the surface and then move over to another cell. So it's really crucial for that motor to be able to be transported from the cell surface to the nucleus where it can propagate and then transport out again to the cell surface. And you can see here in this beautiful movies that Helena has been taking, how those viral particles are moving in a bidirectional manner in the cell center and actually manage to get to the surface of the cell. And we are using that system to try to identify which motors are involved in this process and how the virus is hooking up to those motors. And really all those techniques we kind of aim to use to get to a view that is similar if you go and visit Berlin and you see on the road people going along, walking along, cycling along, going with their cars along. So we try to understand how those motors work, how those microtubers are organized by cross-linking motors, by sliding motors, by cross-linking molecules, how those kinesins that go one way and the dyneins that move the other way are coordinating the transport of cargoes, and how this is all organized so that we can put more and more actual real data in those pictures and less fiction after a while. So they serve a really good purpose to kind of give you an idea of how we envision how this thing works. And I think with that, I'm starting to come towards the end. So yes, so this is another example of how we try to convey those messages of what we are doing also to the younger generation, of course, and trying to kind of get an understanding of how those molecules work by kind of rebuilding them using all our favorite toys. And if I'm not in the lab, I thought I should say that because people might wonder if what I'm doing when I'm not working. And there are those times that I'm not working. You usually find me somewhere outside with a map in my hand. And I seem to be quite good at this sometimes as well. We're training my next generation to also become vivid and happy and successful orienteers. So it sometimes works out for them too that they win some medals. What comes next? I don't even know what works next. Yeah, and then sometimes things actually come together, work and fun. Our recent challenge 4 days ago set by Helena and our new seems to be Straube club was to move like a kinesin, and that is really very convenient. You take the figure from your latest paper, you put it on your favorite orienteering map, and then you go and run along. And this is a good way of combining work and joy, at least my form of joy. So I think I start to come to the thanks. I think you have seen those before, but I think this is kind of, yeah, a good moment to say thanks to my super colleagues who have been over the years collaborators, mentors, giving me great advice, brutally honest feedback, which is one of the most valuable things you can get from people, and just generally turning out into being friends. And also external members and collaborators who have really shaped my last, 10, 20 years in science. Bill Earnshaw has been my postdoctoral mentor when I was in Edinburgh, and was also very good at giving brutally honest feedback, which is super useful. Anna I met as a postdoc already, and we really shared interest and mindset. And she has been instrumental in getting my lab going and using in vitro reconstitution. So she hosted me in her lab to teach me how to do that, so I could move that over to my own lab. And then there are some long-term collaborators, Irina, Jenny, Andrew, Richard. Those are the ones who I published more than one paper with. And I think we all tried to also write grants together, which didn't always work out, or maybe most of the time don't work out, but it's still an important part of the journey. And then, of course, there are all those people who worked with me, and some of them still work with me. There are meanwhile 2 slides of those, I realized. I got away at my Lister lecture with 1, but it's now 2 of them. And every one of them has contributed in one way or the other, and we couldn't do anything without them. So thanks to every one of them, and of course the funders, very important to kind of keep us going. I think I might have mentioned them along the line, some of them. That starts from the postdoc fellowship, getting started with my lab, supported by Marie Curie Cancer Care. The Lister Prize was really instrumental in keeping me going at the end of the Marie Curie money. Recent funding, very generous from the Wellcome and the Leverhulme Trust, those for funding PhD studentships, and the British Heart Foundation as well. And I think with that, I'm done. Is anybody still here?
Thank you. So I think for this, it'll be lovely if we had the microphone for the real applause. But anyway, yeah, we did all right for the real applause. So thank you, Anne, for that brilliant lecture. I think, first of all, we understood a bit more about your own personal journey, and that itself is impressive. But in terms of the actual lecture, you know, this is not an area I know very deeply, but I couldn't be impressed by the huge commitment to understanding deeply what happens in the cell. Because I don't know about you, but certainly I've had close people succumb to cancer, for instance. And one of the areas that where we need to make better progress is to actually fundamentally understand how things normally work and what goes wrong. Because without that, we can't get to better treatments with less toxicity, etc. So I think the work you're doing is clearly hugely important. So it's important that you continue, and hopefully it takes a long time to go from there to get a new treatment, but hopefully this is what will get us further forward. So thank you again very much. Lovely visuals, by the way, very impressive slides and so on. So, and I learned a few things, even though I'm not an expert in this field, and I'm sure those who have listened may have got even more out of it. So without much further ado, I just want to say thank you once again for leading the charge with the Leading Lights lecture this year. And I'd like to invite all of you to attend the other lectures during this year where we celebrate the Warwick Medical School's 20th birthday. So now I think you can have lunch, but sadly, individually, I think we can't go off and have lunch with family like we normally do. But once again, I want to say thank you and congratulations.
Thank you. Thank you all for coming.
Bye then.
Bye.
All right. Bye.